Viruses, those enigmatic entities straddling the line between living and non-living, boast an ancient lineage, far surpassing the reign of dinosaurs. These microscopic agents have been around for billions of years, long before the first dinosaur roamed the Earth. Their existence predates even the earliest forms of cellular life, making them one of the most ancient forms of biological entities known to science. Their origins remain shrouded in mystery, with several compelling hypotheses vying for acceptance. Scientists have long debated the true beginnings of these elusive entities, and despite significant advancements in molecular biology and genetics, the question of where viruses come from remains one of the most intriguing puzzles in the field of evolutionary biology.
One theory posits that viruses represent ancient life forms, predating the emergence of cellular life. This hypothesis suggests that viruses could be remnants of a pre-cellular world, a time when life was just beginning to take shape in the form of self-replicating molecules. Imagine a primordial soup teeming with self-replicating molecules, some of which evolved into the complex machinery of cells, while others persisted as simpler parasitic entities, the precursors to modern-day viruses. In this ancient prebiotic environment, these primitive entities might have played a crucial role in the early stages of life's evolution, acting as catalysts for the development of more complex biological systems.
Another hypothesis proposes that viruses originated from fragments of cellular genetic material that gained the ability to exist and replicate independently. This theory, known as the escape hypothesis, suggests that viruses are essentially rogue pieces of genetic material that broke away from their original cellular hosts. Picture a cellular escape artist, a piece of DNA or RNA breaking free from its confines, acquiring the tools to hijack the machinery of other cells. These genetic escapees would have evolved mechanisms to enter host cells, replicate, and spread, giving rise to the diverse array of viruses we see today.
Regardless of their exact beginnings, one thing is certain: viruses have been shaping life on Earth for eons. From the earliest days of life on our planet, viruses have been a driving force in the evolution of living organisms, influencing genetic diversity and adaptation. Their evolutionary journey intertwined with our own. The relationship between viruses and their hosts is a complex and dynamic one, with viruses often driving evolutionary change through their interactions with cellular life forms. Their genetic fingerprints can be found embedded within the genomes of every life form, from bacteria to humans. These viral elements, known as endogenous viral elements, are remnants of ancient viral infections that have become a permanent part of the host's genetic material, a testament to their pervasive presence throughout evolutionary history. Fossil records and genetic studies reveal that viruses have been a constant presence, influencing the course of evolution and the development of life on Earth.
These viral remnants, often referred to as fossil viruses, provide a glimpse into the deep past. By studying these ancient viral sequences, scientists can gain insights into the long and complex history of viral evolution, allowing scientists to trace the ancient interactions between viruses and their hosts. This research helps us understand how viruses have co-evolved with their hosts over millions of years, shaping the genetic landscape of life on Earth. Understanding the origins of viruses is not merely an academic exercise. It holds the key to unlocking the secrets of viral evolution and their remarkable ability to adapt and persist in an ever-changing world. By unraveling the mysteries of viral origins, we can better understand the fundamental processes that drive evolution and the intricate relationships between viruses and their hosts. This knowledge is crucial for developing new strategies to combat viral diseases and harnessing the potential of viruses for beneficial purposes, such as gene therapy and biotechnology.
Unlike cellular life, which reproduces through processes like cell division, where a single cell splits into two identical daughter cells, viruses replicate through a cycle of hijacking and assembly. This process is vastly different and more complex than simple cell division. They lack the necessary machinery to replicate independently, and instead rely on the cellular machinery of their hosts. Without a host, a virus is essentially inert, unable to carry out any life-sustaining functions. This dependence on host cells for replication is a defining characteristic of viruses, setting them apart from other life forms. Unlike bacteria, which can thrive and reproduce on their own, viruses are more like biological parasites. The viral replication cycle begins with a virus attaching to a host cell, a process known as adsorption. The virus identifies and binds to specific receptors on the surface of the host cell. Its genetic material then slips through the cell's defenses like a microscopic Trojan horse, a process called penetration. This genetic material can be either DNA or RNA, depending on the type of virus. Once inside, the viral genome commandeers the host cell's machinery, forcing it to churn out copies of the viral proteins and genetic material. This stage is known as biosynthesis. Imagine a factory retooled overnight, its production line now dedicated to assembling an army of invaders. The host cell's resources are diverted to produce viral components instead of its own. These newly minted viral components then assemble into new viral particles, a process called maturation. Each component fits together like pieces of a puzzle, forming countless copies of the original intruder. Countless copies of the original intruder are now ready to continue the cycle of infection. The host cell becomes a ticking time bomb filled with new viruses. The host cell, its resources depleted and its machinery overwhelmed, often succumbs to the infection, releasing a flood of new viruses to infect neighboring cells. This final stage is known as lysis. This cycle of infection, replication, and release drives the rapid evolution of viruses. Each new generation of viruses can potentially carry mutations, leading to new strains that may be more virulent or resistant to treatments. Understanding this complex cycle is crucial for developing effective antiviral drugs and treatments. By targeting specific stages of the viral replication process, scientists hope to disrupt the cycle and prevent the spread of infections. For instance, some antiviral drugs aim to block the virus from attaching to host cells, while others interfere with the replication of viral genetic material. Each approach offers a potential pathway to combat viral infections. As we continue to study viruses and their replication cycles, we gain valuable insights into how to control and prevent viral outbreaks. This knowledge is essential for protecting public health and combating future pandemics. The battle against viruses is ongoing, but with each discovery we move closer to understanding and ultimately overcoming these microscopic invaders.
Viruses exhibit remarkable genetic diversity, a consequence of their rapid replication and error-prone copying mechanisms. This diversity is particularly pronounced among RNA viruses, whose genomes are composed of ribonucleic acid rather than deoxyribonucleic acid, or DNA. RNA, a single-stranded molecule, is inherently less stable than DNA and more prone to errors during replication.
These errors, known as mutations, introduce changes in the viral genome, leading to variations in viral proteins and, consequently, viral characteristics. Imagine a blueprint with typos, each error potentially altering the final product.
Some mutations might be detrimental, rendering the virus unable to replicate or infect cells effectively. Others, however, might confer an advantage, allowing the virus to evade the host's immune system, spread more efficiently, or infect new types of cells. This constant generation of genetic diversity is a hallmark of RNA viruses, enabling them to rapidly adapt to changing environments and challenges, including the defenses mounted by their hosts.
The relentless pressure exerted by the host's immune system and other environmental factors acts as a sieve, selecting for viruses that can survive and reproduce most effectively. This means that only the viruses with the right traits to overcome these challenges can continue to thrive. It's a constant battle, with the immune system trying to eliminate the virus, while the virus evolves to evade detection and destruction. This process, known as natural selection, is the driving force behind viral evolution. Natural selection ensures that the most adaptable and resilient viruses are the ones that persist. Over time, this leads to the development of viral populations that are better suited to their environments, whether that means evading the immune system or finding new ways to infect host cells.
Imagine a race with constantly shifting finish lines, where only the swiftest and most adaptable competitors can keep pace. In this race, the finish lines represent the various challenges and obstacles that viruses must overcome to survive. These obstacles can include the host's immune response, antiviral drugs, and even changes in the environment. The viruses that can quickly adapt to these shifting challenges are the ones that succeed. Viruses carrying beneficial mutations, such as those that enhance their ability to evade the immune system or infect new cell types, are more likely to survive and reproduce, passing these advantageous traits to their progeny. These mutations can give the virus a significant advantage, allowing it to spread more effectively and potentially cause more severe infections. As these beneficial traits are passed down, the viral population becomes more adept at surviving and thriving in its environment.
Over time, these beneficial mutations can become widespread within a viral population, leading to the emergence of new viral strains with altered characteristics. These new strains may have different properties, such as increased transmissibility or resistance to antiviral treatments. This continuous process of mutation and selection drives the evolution of viruses, making them ever more challenging to control and treat. This process of adaptation is continuous, with viruses constantly evolving in response to the selective pressures they encounter. Every time a virus replicates, there is a chance for mutations to occur. Some of these mutations may be neutral or even harmful, but others can provide a survival advantage. The constant interplay between viral evolution and the host's defenses creates a dynamic and ever-changing landscape.
The ability of viruses to rapidly adapt through natural selection has profound implications for our understanding of viral emergence, disease outbreaks, and the development of effective antiviral therapies. By studying how viruses evolve, scientists can better predict which strains might become more prevalent and develop strategies to counteract them. This knowledge is crucial for designing vaccines and treatments that can keep up with the ever-evolving viral threats. Understanding the mechanisms of viral adaptation also helps in anticipating potential future pandemics and preparing more effective public health responses.